Hello and welcome to Delve Deeper, the MASTS 2024 webinar that delves so deep that even Cuvier's beaked whale was shocked to see us. We have an absolutely pearl of a presentation for all you shell seekers in the audience today, as we explore the fascinating world of molluscan biology in today's talk, from pearl formation to shellfish reefs, applying molecular techniques to malacological questions. So without further delay, let's introduce our esteemed speaker of the day. Our speaker is a leading researcher in comparative and functional genomics, driving advancements in sustainable aquaculture and marine conservation. Their expertise spans evolutionary biology, focusing on genetic mechanisms in mollusks and annelids, molecular aquaculture for enhanced pearl quality and oyster production, and genetic identification techniques for biodiversity assessment in shellfish reef restoration. Please give a warm virtual welcome for Dr. Carmen McDougall.
Thanks very much, Phil, for that introduction, and sorry about the tongue twister of a title. It's quite broad, the different areas that I'm trying to cover today, but I'm basically trying to demonstrate the utility of molecular tools for understanding basic molluscan biology. So I hope you enjoy what I have for you today. All right, just before I get started on my presentation proper, a lot of the work I'm going to be talking about today was conducted in Australia, and accordingly I'd like to acknowledge the traditional custodians of the lands on which this research was conducted and to pay my respects to their elders past and present. Just going to slide. There we go. So just a little bit about me. You might gather from my accent that I'm not Scottish. I'm from Australia, so I did my undergraduate at the University of Queensland before moving over to the UK to do my PhD at the University of Oxford. But I actually spent the last year of that PhD at the University of St Andrews, where I am now. At the end of my PhD, I travelled back to Australia and started a postdoctoral fellowship that actually lasted for 9 years at the University of Queensland, and that was working on pearl formation. So I'll be covering a little bit of that research today. After that, I got an independent academic position at Griffith University before moving back to the university in St Andrews early last year, and I've been here for about a year so far. My research group has quite broad interests. So at my core, I am fascinated by evolution and development. So I'm interested in questions about biomineralization, body plan evolution, and self non-self recognition comes into this as well. And that's predominantly in mollusks that I'm looking at these questions, but I do a little bit of annelid work there as well. I'm interested, and my research group is interested in aquaculture, particularly of molluscan species. And more recently, I've been starting to get into molecular ecology. So I'm going to be talking about some biomineralization research, moving into pearl research within the aquaculture space, and finishing off with some molecular ecology around shellfish reefs. So we'll kick off with molluscan biomineralization. And I hope that most of you are familiar with molluscan shells, and so being good marine scientists, you won't be able to walk along a beach without picking one of these up, but you may not appreciate just how stunning they are at the microscopic scale or how intricate. So the molluscan shell is generally composed of multiple layers of calcium carbonate, and each of these is composed of calcium carbonate tablets, and these tablets are actually surrounded by and perfused by an organic matrix, which is made of proteins, polysaccharides, and lipids as well, we believe. And so this organic matrix is thought to dictate the actual structure of the shell and these different layers that we can see. And you'll see here in this cutaway section, we've got a prismatic layer on the top and a nacreous layer at the bottom. And these are different because their tablets of calcium carbonate have different morphologies, but also the calcium carbonate is made up of a different polymorph, so a different crystal structure essentially. And so here we have the prismatic layer, which is actually made up of calcite, and the nacreous layer is made up of a different polymorph, which is aragonite. So these shells can actually be really quite intricate in their detail. The shell I've shown here is kind of our typical model species shell, where we just have two layers, this outer prismatic layer and the inner nacreous layer, which gives many shells that beautiful mother of pearl coloration that you'll see. But other shells from different species have multiple layers with really varied textures. And so prismatic and nacre are just two examples of these. So that's the shell itself, but what we need to remember is that the shell is actually secreted by a living animal. And so here we've got a cutaway cross-section of a shell, this typical two-layered shell that I was talking about. So we've got this prismatic layer, which is the outer layer of the shell. So this would be the shell surface at the top. And we've got the nacreous layer just lying underneath that prismatic layer. But this shell is actually lying on top of a piece of tissue, which is the actual organism. And this organ is responsible for secreting the shell, and this is called the mantle. All right, so it's this mantle, and particularly these epithelial cells that are at the surface of the mantle, which are responsible for secreting this organic matrix, which therefore and then controls the deposition of the shell and the shell characteristics that I was talking to you about before. And some of the really early work that I did was looking at biomineralization in the tropical abalone. So this is an example. The juveniles are really quite beautiful. We're looking at the underside of one of these here. It's less than a centimetre across at this stage. It's probably a couple of millimetres, this one. But this is the underside, and you might be able to see the beginnings of a little bit of a sheen here. So that's the nacreous layer that's just starting to be deposited and thicken. But at the edge here, I think you can see that the shell looks a little bit different, and that's where we're seeing the prismatic layer that actually is found on the outside of most of the shell, but tends to be secreted first at this leading edge. And some of the work that I was involved with, we were identifying genes that were expressed in this mantle tissue and having a look at where they were expressed within the mantle itself. And so we found that these genes tended to be expressed in different zones. So in these images here, we're looking down at the surface of the animal. Okay, so the shell would normally sit on top of this animal here, and where you can, this is one mantle lobe on this side, and then the other mantle lobe is over here. So normally that would be sitting right underneath the shell. And we can do a technique called in situ hybridization, which stains up in blue where this gene is expressed in the mantle. And what I hope you can see here is that we get blue staining in different regions. So in these first panels, you can see that we've got fairly uniform staining, but right at the very edge of the mantle. We've got other patterns that seem a little bit more blotchy, have a bit more of intricate patterning. And then we have other expression patterns that are much, I guess, step back a little bit from the shell edge and have a broader expression pattern than those ones before. And then we have expression patterns that are much more towards, I guess, the center of the mantle. And so what we know is that these genes encode proteins that can be found in the different shell layers. And we know now that this gene here, which we call somatske, this gene is actually responsible for creating this red pigment that you can see in the outside layer of the shell, and it's expressed in this zone. So we know that this region of the mantle produces the prismatic layer. And then further, we know that these two genes encode proteins that are found in the nacreous layer of the shell. So we know that this region of the mantle is responsible for secreting the proteins that are involved in nacre production. And so this really showed us that the mantle is regionalized to an extent that we didn't understand before. And we now know that this regionalization can be found broadly across different mollusks, so both gastropods and bivalve mollusks. And we can therefore infer that this regionalization of the mantle is responsible for dictating shell morphology across bivalves and mollusks. But for me, the question about how these zones are set up in the first place and how changes in that process have led to the evolution of the diversity of the shells that we observe today, that's where the interesting question is, and we still really don't have answers to those. So just thinking about this further, I'm interested in the regionalization of this mantle by mineralization gene network and how all of this is regulated at the genetic level. And I'm also interested in what are the differences in those proteins that facilitate the formation of the different ultrastructures and polymorphs that we see in different species. And so work that's ongoing at the moment is looking at this in a bivalve this time. So this is the pearl oyster punctata maxima. This is the shell. So you can see it's got a very famous mother of pearl inner nacreous layer and an outer prismatic layer. And so here we have a cross section through the mantle tissue, and we've done micro dissections to dissect out each individual piece of this mantle. We've sequenced all the genes that are expressed in each of these pieces, and we're correlating those back to the shell itself. What we're going to do then, and we're currently in the process of it, is using modeling and bioinformatics to try and find out what genes are, I guess, most connected within these gene networks. And these are probably the genes that are going to be responsible for I guess, setting up this entire gene regulatory network and controlling what layer forms where. So that's work that's still in progress, but hopefully I'll have some answers for you about that soon. And this is actually a nice segue into what I wanted to talk about next, which is understanding pearl formation. So many of you will be familiar with pearls, but may not be very familiar with how they're formed. So natural pearls are actually very rare in nature, and what you see mostly found in jewelry are the result of pearl culture. And so when a cultured pearl is made, what we have first is a donor oyster, and this oyster is normally selected because of the really beautiful nacre that it's producing. So we want something that's, I guess, the color of the pearl that you want to produce. That the mantle that produces that nacre is then dissected out and used as a graft in a second oyster. So over here you can see we've got a second pearl oyster on a stand. It's a bit hard to see because the light's in the way, unfortunately, but it's open and is currently being operated on. So the technician will make a cut within the gonad of this of this host oyster, will then insert a nucleus, which is a spherical bead, into that cut, and will then put a piece of this mantle tissue from the other oyster in behind that nucleus. And what's really interesting there is that the tissue from this piece of mantle will then grow around the nucleus, and it will start producing a prismatic layer, and then it will start producing a nacreous layer. And so you can kind of think of a pearl as a bit of an inverted shell in that way. It's a little bit more complex, but we won't go into those details there. And so ideally what you have after putting this oyster back into the ocean and letting it grow for about 2 years is a beautiful, nice, spherical, lustrous pearl that's going to get you lots of money. But it's not always the case. So with pearl formation, this is what we're aiming for, what you can see in panel A here. But pearls often have a variety of different defects. So this one is quite dull. It's not quite as shiny as this first one. So we would say that this has high luster and this would have low luster. We can also have this defect where instead of the nice shiny nacre, we kind of get this opaque appearance. And in the industry, this is called calcification, which is a bit of a misnomer because obviously the entire pearl is calcified. But this can either occur in broad patches of the pearl or in tiny spots on the surface. And work done by geochemists has actually showed us that this kind of cloudiness comes from the formation of another polymorph of calcium carbonate, which is called vaterite. So that's an interesting one. And I won't go into all these different defects in the bottom, but you can see we have different surface defects as well. And so what we did, we're interested in understanding what might be causing these different defects. So I went along to a pearl harvest and watched what was coming out, and we selected 28 pearls and their matching pearl sacs. So that's the tissue that actually secreted the pearl, and harvested them at this time. And those pearls had a range of different characteristics or defects according to the ones that I've showed you here. We then got the pearls graded professionally by a pearl grader, so we knew exactly what class they would be. And we also took those pearl sacs and extracted the RNA from them and did transcriptomics and differential gene expression so we could understand what genes were differentially expressed between pearls that had or did not have these different defects. And I'll go over these results quite quickly because I know heat maps scare a lot of people. But basically, each column is a different pearl sac. Each row is a different gene. Okay, and so the pearls that had high luster, so the nice shiny ones, are grouped over on the left-hand side of this chart, and those that had duller luster were on the right-hand side. And I hope you can see that we've got more red colors for these genes in the high luster pearls, whereas we've got more blue colors here. And that's an indication of the level of gene expression. So these genes tended to be more highly expressed in higher luster pearls, whereas these genes were more lowly expressed. And when we looked at what these genes actually were, we found out that most of them were genes that encoded known shell matrix proteins. So the proteins were actually secreted into the organic matrix and controlled shell formation. So it looks like luster is associated with the level of expression of these genes. So that might indicate that I guess the pearl sacs are more transcriptionally active when we've got nice lustrous pearls. So that actually wasn't too much of a surprise. Our surprising result came from the pearls that had this calcification defect. So we found a really large number. This is just a subset of the genes that were differentially expressed between calcified pearls, which are over here on the right-hand side, and pearls that didn't show this calcification defect. And when we looked into what these actually were, we got a bit of a surprise because many of these genes were actually annotated as being involved in cilia formation. And that seemed a little bit odd. So we had a bit more of a look into this and did some tests, and it's not due to sperm contamination or something like this. It looks like we do have a real signal here. And so that's indicating to us that cilia might actually play a role in polymorph control in this species. So that's certainly something that we need to look into a little bit further. So I think this shows you that using molecular techniques can really help with some functional understanding of biomineralization. But I'm also interested in the evolution. So here we have a phylogenetic tree of the different classes of mollusks. And so far, we've looked at biomineralization most comprehensively in these two. Okay, so in the gastropods and the bivalves. And when we compare the shell matrix genes that we find in both of these groups, we find very few shared across the different classes of mollusks and indeed between different groups of mollusks within each of these classes. And this big difference that we see between the gastropods and bivalves may be due to the fact that shells are thought to have originated independently in these two different molluscan classes. So that's interesting, but what is also interesting to look at is what's going on in the rest of the mollusks. We're still not there yet, and hopefully we can answer some of the questions on that shortly as well. All right, so I'm going to leave the biomineralization aside for the moment and jump into something completely different, which is understanding oyster biodiversity. So when I was doing my postdoc, I got really interested in molluscan aquaculture. And then when I started at Griffith University with my new fellowship, I was transferring some of that knowledge to the rock oyster industry in Australia. And this lease here basically shows you a mostly empty lease. And that's because of issues that they're having with disease, with rock oyster aquaculture within especially northern Eastern Australia. And all the baskets here are just holding the experimental animals for one of my PhD students. So the situation is really not good. And so one of the, I guess, industry's responses to this is to ask whether or not there are other species that naturally exist along the coast that we could be using for aquaculture instead, because there are certainly oysters there. And so we just didn't know what they were. So my project was quite a nice one where I got to travel up and down the Queensland coast. So this is about, I think, 2,000 kilometres in total. Basically sampling oysters and sequencing them to find out what species they were, because if you know anything about oysters, it's pretty hard to distinguish what species they are just by looking at their morphology. And to our surprise, we found that we had 14 different lineages. This hopefully will come out very soon. And you can see this guide to intertidal oysters. That's available if you're interested in exactly which ones we found. But among these 14, there are a couple of species in particular that are quite interesting for aquaculture. And annoyingly, in order to do some spawning and hatchery trials, we needed a way to non-invasively figure out what species we were dealing with. So this is Marina, my PhD student, trying to swab an oyster here so that we could genetically determine which actual species it was. But the exciting thing is that colleagues at the Department of Agriculture and Fisheries are currently doing spawning trials with several of these species, and they're having really good success. So that's one aspect to determine what species we are and determine whether or not we can produce these in a hatchery and whether they grow well. But in Australia in particular, we have a lot of questions around where we should be translocating individuals, even if they're from the same species. So we have a question about genetic variation between populations in Australia. And this is some work that was done by colleagues in the Northern Territory, which is up here, for one of the species. And they basically did population genetics on a range of populations around the Northern Territory, but also included one Queensland population here in Bowen and in Noumea as well. And so this MDS plot basically shows you that the Northern Territory ones, even within this location, we can see two pretty distinct genetic populations, which is interesting. These appear to be different to Bowen and completely separate to the New Caledonian specimens as well. So we've got work underway at the moment to expand this to multiple locations along the Queensland coast and to really try and get a better understanding of the genetic diversity across populations in all these different areas. This work was very fun, and it ended up leading me in a completely different research direction altogether, which is shellfish reef restoration. And so some of you may be aware of the demise of shellfish reefs around the world. There's been a lot published on this. So oyster reefs have declined globally by about 85%. And so there really has been almost a worldwide restoration movement to recover the lost ecosystem services that these shellfish reefs provided. When we started this project, we were quite struck by the fact that the global assessments and most restoration projects have occurred most exclusively in temperate and subtropical regions. But we know that oyster reefs historically have also occurred throughout the tropics. So my PhD student, Marina, who I showed you just before, did a literature review to try and understand the global diversity of reef-building oysters. And so this review essentially highlighted that we did have 38 species of oysters that were known to be reef-building globally. And of these, 25 of them were tropical species. So this is showing us that tropical regions contain over 4 times more reef-building species than temperate regions. There are also some other differences between tropical shellfish reefs and temperate ones. In tropical regions, we tend to have spatfall throughout the year, whereas in temperate regions, these tend to be a lot more seasonal. Tropical oysters tend to be faster growing than temperate ones. And tropical oysters also commonly create mixed species reefs, whereas our temperate shellfish reefs tend to be dominated by a single species. So these are all differences between tropical and temperate shellfish reefs that need to be taken into account when designing a restoration project in the tropics. And this was all really interesting, and we'd heard all this doom and gloom about shellfish reefs and how they pretty much all being wiped out. So we were pretty surprised in our oyster survey when we came across this really healthy looking shellfish reef near Gladstone in Queensland. And that triggered us off on a bit more of a search, and we've found since then additional very healthy tropical shellfish reefs in other regions. And so Marina's PhD project took on a little bit of a different direction from shellfish reef restoration to characterization of existing healthy shellfish reefs. And so this just shows you some drone footage of one of these reefs. This is the one near Gladstone. You can see it's quite extensive, but it's also existing on a really muddy site. And so this is interesting because these oyster reefs are not, they're kind of self-sustaining in a way. So these oysters are not settling on rocks. They're actually creating the substrate themselves in this environment, which is pretty interesting. And this mud is really squelchy mud. So if you stand on it, you disappear almost to your waist, as we've found out while trying to sample these things. So Marina's busily trying to finish off her PhD at the moment within the next couple of months, but we've got some preliminary results from Gladstone. So by looking at this drone footage, we can summarise that we've got about 4.2 hectares of oyster reefs with over 100,000 individual reef patches. They're all quite high profile, so emerging more than 15 centimetres from the substrate. They've got very high density of oysters, and that this primary settlement substrate is soft sediment and shell, which is interesting. And our preliminary results when comparing this with the other reefs that we've found in the regions is that the other two are actually on rock, but this soft sediment shellfish reef seems to harbour greater biodiversity of the invertebrates that live on the reef than the ones in the other regions, which is interesting. But we're following up on research in that area and should be able to present a more thorough assessment very soon on all of this. So I think I am out of my designated time, but I'd just like to acknowledge everyone who helped with this study. So students who have worked in the lab on this project, collaborators and partners. Thank you very much, and I'm happy to take any questions you might have.
Got to unmute myself. Thank you so much. That was really incredible. You've had such a wonderful career. That was really nice to see sort of the stages you've gone through. I was originally my kind of original first kind of question I was thinking of was actually to do with the pearl formation and kind of because I know that there it is an incredibly huge industry, but my kind of I've always wondered what actual pearls are used for by the animals that make them. And but from the sounds of your presentation, it's more of an accident.
Yeah, so naturally forming pearls, and there's the myth about a grain of sand and it irritates the oyster and it secretes around a grain of sand. That hasn't, there have been studies where people have used, I think, CT scanning and X-ray tomography to try and find out what's at the center of these natural pearls, and it's never a grain of sand. But often it just seems to be that, and we're still working on this, something happens which dislodges some cells from the mantle and into the body of the animal, which shouldn't normally happen. And that triggers a shell formation response in that region of the animal. So then you start getting this spherical pearl forming in a place that you shouldn't have shell secretion at all. So some of it's probably a defense mechanism as well against shell borers. So you'll often see, and on the farm, I think one of their greatest annoyances is the polychaete worm. That's a boring worm that's trying to drill into the shell from the outside. So they spend a lot of time trying to protect the oysters from those worms. But if you can see that one has actually gotten inside, it'll start creating what they call a blister pearl on the inside. And that's basically a shell repair mechanism. So it's a raised surface on the inside of the shell. So that can be another mechanism as well.
I actually generally did think it was the whole a grain of sand. It's one of those like I heard it and it seemed to add up, so I never really questioned it.
Yeah.
So we are officially in the sort of discussion Q&A section. Just to let everyone know in our audience, you can use the raise hand function if you'd like. You may need to give me a couple of seconds just to unmute you. Otherwise, you can use the Q&A section, which I'm just going to open just in case someone is writing questions in there. But I do have a question to sort of start us off. And it kind of goes back to what you were saying about this huge decline in shellfish reefs. and kind of also goes back to your work in molecular tools and sort of the understanding molluscan molecular biology. Are there any tools that you think can play a role in restoration and management of these marine habitats to try and help restore them? Or is there at least a tool that maybe suggests when these reefs are potentially going to decline rapidly? Is that something that you've come across?
Molecular tools, you mean?
Yes,
Yeah.
yes.
Yeah, I think the idea of having a predictive one for decline is interesting, and I can't think of how you would do that out of the top of your head. But I think in terms of aiding restoration, it's really important to understand the genetic diversity of existing populations and also what you're restoring. And I know there's a big debate about whether or not hatchery spat should be used for restoration purposes. And the concern is that it may decrease the genetic diversity that you have in these areas. So there's a lot of interest in that area. So doing some kind of general population genetics, I think, is really important for conservation. I think as well, and it's early stages, but when you are restoring shellfish reefs, it's important to consider why the shellfish reefs may have declined in the first place. So if that stressor still exists in that environment, you might need to have more resilient oysters, if it's an oyster reef restoration project, in order to overcome that. So some kind of selective breeding might be important there as well.
Okay. So, and again, going back to the actual biomineralization. So, and this might just be so I once upon a time did some research in ocean acidification on marine worms. So I have a rough understanding of how ocean acidification might be interrupting things like calcification in certain animals. Is that potentially a pathway that you see that's going to have a real big impact on I always struggle to say this word, biomineralization,
Yeah.
or is this something that the species you looked at were able to kind of cope with quite well?
I mean, there's been a lot of studies looking at the effects of ocean acidification on biomineralization, and there's really conflicting results depending on what species you're looking at and also what life stage. So the larvae do tend to be a lot more susceptible than adults are. So that might be an argument for hatchery reared larvae, because then at least you're controlling the conditions in your hatchery before putting these animals out in the real world. It's certainly an important question to look at, and it would be also interesting to know if you could breed for resilience to that as well, because yeah, certainly some populations are likely to be more resilient than others.
So I potentially have a follow-up, but we are getting some questions in from the audience, so I don't
Yep.
want to take up all the time. Firstly, we're getting some really nice feedback for your talk, which is always nice to see. We have one person that's asked, do you think there's a possibility of creating a hybridized, they've SPP, but it could be app, a hybridized SPP or app that might enable
Species, I think,
greater
yeah.
SPV, that makes sense. Hybridized SPV that might enable greater settlement of oyster reefs in areas where there's little to none hard substrate.
Yeah, I think that's a interesting question, and we are interested as to whether or not you could create viable hybrids for these species. It's really interesting that they all live in, they're all sympatric. They all live in the same environment, but somehow maintain their genetic distinctiveness. So that might argue against the natural hybridization of these species. But I think, yeah, that is certainly interesting to look at. So the species that were settling on the reefs are actually one of our, I guess, unnamed lineages, and I didn't get into all of that, but oyster taxonomy is a little bit of a mess. But it's the one that creates these crazy vertical spines out of its surface when they're young. So their shells actually have these spines on them. So it was that species that are creating that particular reef. And it would be really interesting to look at whether or not we can create hybrids and whether that would then, yes, change the settlement preferences of these species. But yeah, a lot of work to do to answer those questions.
Seems like there are a few unknowns when working with these. Well, I mean, it's one of those sea creatures that always just baffles me because you look at it and you don't necessarily see a lot, but there is
Mm.
so much to them.
Hmm.
Um,
Traditionally
so
overlooked, and it's funny going to museum collections and there's very few oysters in there, so plenty of pretty
this,
mollusks.
yeah, well, this is like again kind of why I've been really interested in this talk because I do have a history in marine biology, but I've never spent a lot of time looking into these species. So this has been a really interesting talk. Um, I've got another question here. I'm gonna so uh, it was noticeable that the in speech marks lustra genes had a lot more unknowns as well as some oddly named genes, including mammalian and venom. Is it simply due to relatively poor annotation of molluscous genomes, or does this reflect the biology and the idea of biomineralization genes are evolving quickly and independently in different groups? So these genes are taxonomically restricted genes.
Yeah, that's a good observation. And I think it's a bit of both. So definitely annotating, and anybody who's working with mollusks will know that doing your usual annotations based on blast homology will give you pretty much 50% unknown genes. So that's something as a community I think we all need to work on. But we also, from some of the work that we've done, noticed that a lot of these genes that make up the shell matrix are rife with repetitive domains and just crazy sequences. So they're probably quite fast evolving, and that will also make it very different to, sorry, I'm trying to read the full question. I think that's from Dave. Yeah, really hard to understand how these things have evolved or detect homology. So I think it's a question of both.
That was quite an in-depth question, actually. Brings up a lot of interesting thoughts. I think I've only really understood the last part of it. In all honesty, I think I might need to reread that question. But again,
Mhm.
really, really interesting. We do have another question, and we still got time. So do we know sorry, let me try that again. Do we know much about the influence of genetics and environmental conditions on shell formation, particularly influences on shell strength in mussels?
Hi, Greg. Yeah, really good question. Um, there are trials that are underway. I don't know if in mussels, but on how things like salinity can affect shell properties, and there is evidence that it can. I can't remember, I've just read it, but I can't remember the paper off the top of my head. But yes, we know that there is an influence, but I think it's pretty complex to tease out the interactions between different environmental conditions on things like shell strength. And then genetics will come into it as well. So it's a pretty complex question. Um, but I know people are looking at it. And yes, I would say yes, there are influences both ways. I hope that answers your question.
So I think we have time for maybe one more question or potentially two. So if anyone does have oh, we've had one just come in there. I'm looking at the wrong end. So regarding museum collections, are paleontological collections too old to study because there's often masses of oysters and other shells in
Mhm.
there?
Um,
Maybe less of a question than an explanation, but
yeah, no, I think that's really interesting. Um, there is a really interesting movement in ancient DNA. So there's several groups. There's one, I think, in Denmark, and I'm trying to think the oldest shell they've gotten DNA out of. I think it's something like 1,000 years. It's pretty cool. And I know there's a group in New Zealand as well that are going to museum specimens, grinding out a little bit of the shell um, and getting sequenced that way to do species identification and things. So I think you can. The DNA is obviously not in great shape. Sometimes it's good enough to do, molecular species identification. I think it would be awesome if we could use that to answer other questions, more evolutionary questions, and you could start trying to find the genes that, a shell matrix protein genes, and look at how these evolve over time. But I'm just not sure the DNA will be in a good enough state for that. But yes, depending on how old they are, I think we can get molecular information out of them.
So I'm just going to double check if, because I don't always get notifications when questions come in, but I think that's all the questions we've had so far. I do have one more question, and I'm not going to claim this to be my question, actually. So I often use AI to help generate text for our website and to sort of get things ready for these webinars and events. And I just decided on a whim, just as I'd input some of the summary from your from the event online into it, whether or not I could ask Chat GPT to ask you a question.
Okay.
And it actually came up with something that was pretty good. So the question was, how do you envision the future integration of emerging molecular technologies, such as single-cell genomics and epigenetics, in advancing our understanding of complex biological processes in mollusks and their ecosystem? Which
Interesting.
potentially a bit
Thanks,
of a mean question as well. But this idea of where is the future with this research?
Yeah.
What could be the potential future implications of what we're doing now?
And I think that's really interesting. And there's several components to that, because if we're talking about ecology and environment, and probably other aspects as well, but I think epigenetics is really critical for understanding things like transgenerational inheritance of resilience to stresses and things like that. It's early days for that kind of research in mollusks, but I think it's certainly going to make some really interesting insights as to how these animals are actually adapting and what capacity they have to adapt to future climate change scenarios. So that's that aspect, and that's, I guess, the broad conservation aspect and environmental. But then there's also drilling right down into fundamental cell biology of mollusks and this single cell sequencing, for things like understanding this regionalization of the mantle. Even by doing this gene expression staining that I showed you before, that only gives you a really broad indication of, there's some cells in this region that are expressing these genes, but then there's not going to be a single cell type. So it'll be interesting to have a look at all the different cell types that are involved in biomineralization, how they develop over the life cycle of individual mollusk, and also how evolutionary changes at the cellular level have led to the different shell morphologies that we see. So that's my answer to Chat GPT.
It's one of the questions, again, I think with everything happening right now, RE, climate change and ocean acidification, it is one of those kind of subjects that's always in the back of my mind whenever I'm reading through research, is what now, as we get to a more of a kind of a scarier kind of idea, scarier future of these big figures and big numbers and big researches coming out, like what are these future ramifications? And I think that's kind of a nice question to end on as well, because I think, research, we're always pushing forward. And on that note, I am gonna end things off. Thank you so much for your talk today. I've had a really amazing time, actually, learning about these animals. Again, it's an area that I just haven't found myself into quite often naturally. So it's been so nice to actually dive into this subject a bit more. I'm also going to thank all of our guests for coming today and all of our attendees. I am going to post into the comments now a link to the MASTS mailing list and newsletter. We will be hosting more webinars just after Easter, plus many other webinar events of our members. So if you're interested in finding out more about things like this, feel free to sign up to that newsletter if you haven't already. And on that note, I am going to end the recording, and thank you all again for joining us.
Thanks, everyone.